Hello everyone, welcome to the biology video lecture. In this video lecture, we are going to discuss the point of a point of cell cycle and cell division, and the division that we are going to discuss over here is meiosis. Okay, so in the last video lectures, we have discussed about mitosis. Also, we have discussed about the cell cycle, right? So we know now what is a cell cycle. We know what happens during the time of mitosis, which is an equational type of division, right? Now we are going to focus on the next type of division, which is called as meiosis. Okay, and in meiosis, okay, which is a bit complex process than the mitosis, and in meiosis there are two M phases that we are going to discuss. In mitosis, we saw only one M phase, okay, that was divided into prophase, metaphase, anaphase, and telophase. In meiosis, there is meiosis 1, okay, and then there is a meiosis 2. Meiosis 1, which is there, okay, it is called as the heterotypic type of a division. Heterotypic division. And meiosis 2, which is there, it is called as the homotypic division. These are the different names that are given to meiosis 1 and meiosis 2. Basically, when we talked about mitosis, okay, in mitosis we saw that there is an equational division. Okay, if a mother cell has two chromosomes, okay, the two daughter cells which are produced also has two chromosomes each, right? So equal number of chromosomes, equal amount of DNA is maintained in the daughter cells. That is not the case in meiosis. Okay, in meiosis, okay, just rough I'm drawing over here. Okay, we will erase this and then again we will discuss the meiosis in detail. But in meiosis, if the mother cell has two chromosome each, we are talking about chromosomes, right? If it has two chromosome each, okay, that is, it is a pair of chromosome. First is meiosis 1, okay, and then is the meiosis 2. At the end of meiosis 2, four daughter cells are produced, and each daughter cell will be having only one set of the chromosome. That is half the number of chromosomes as compared to the mother cell, right? So this is meiosis, and it is seen that mostly this happens in the reproductive cells during the production of gametes. Okay, gametes, these are basically the sex cells which are very much important during the time of sexual reproduction, because the gametes fuse, the male gametes and the female gametes has to be fused, fertilization has to be taken place, and after the fusion the zygote is formed, which is again diploid in nature. Diploid means it also has pair of chromosomes, right? But gamete, it only has sets of chromosomes, right? So gametes are always haploid in nature, and the zygote which is formed, that is diploid after fertilization. So meiosis, the mother cell is diploid, right? The individual is diploid, and the progeny which has to be produced, that also should be diploid, right? So if the diploid number of chromosomes has to be, if the diploid condition of the chromosome has to be maintained in the offsprings, okay, in the next generation, the gametes has to be haploid. Okay, consider if the mother cell is diploid, okay, and the mother cell is producing diploid gametes, right? So if diploid gametes fuse, when the zygote is formed, it is going to be a tetraploid zygote. That is, it will contain four chromosomes, right? So this is going to be an abnormal condition. To avoid this and to restore the same number of chromosomes in the new generation, the gametes which are produced, they are produced in a haploid form. These haploid gametes, they will fuse, fertilization takes place, and after that the zygote is formed, which is again diploid in nature, and this zygote will produce a complete new individual, okay, with the continuous cell division. So that is just the general things that we are just talking about the meiosis. Now let us start with the meiosis. Meiosis, okay, here the term was basically coined by farmer. The term meiosis was coined by farmer and Moore, okay, and in the year 1905, right? And the division which was there, it was first studied by around, there are 3 to 4 scientists: Van Beneden, Van Beneden. Then there is Strasburger, who studied the division. Okay, then there is Winnie Water, and Sutton. So these are the four scientists who studied the meiosis, who studied the division of meiosis, and the term meiosis was basically given by Farmer and Moore in the year 1905. Okay, we have already studied interphase during the time of mitosis. We studied that the interphase which has G1 phase, S phase, G2 phase. In G1 phase, okay, the DNA amount is same, okay, the chromosome number is same, and the different varieties of proteins are synthesized in the G1 phase. The organelles which are there, that duplicated during the G1 phase. In synthetic phase, okay, what happens is that when we talk about chromosomes, we imagine these structures, right? But in the synthetic phase, the DNA replication takes place, right? So this chromosome has one molecule of DNA. So when the replication takes place, now there are going to be two molecules of DNA, right? So to keep this DNA, okay, on itself, it has to multiply its, or it has to increase its chromatid number. Chromosome number remains same, but the chromatids are duplicated, right? So now when we talk a pair of chromosome during the time of division, we are going to discuss about, we are going to consider chromosomes like this, okay? And in mitosis, when we talked, these pairs which are there, which are called as a homologous pairs of chromosome, in mitosis during the anaphase time, we have to remember that the spindle fibers, they got connected to a single chromosome from both the sides, okay, during the anaphase, that is from this side also and from this side also to the centromere. So the centromere which is there, it got split, okay, and these chromatids, they got separated and they were pulled to the opposite pole. Same happened with this member also, okay, here also the centromere got split, and because of that, the one chromatid went on this side and the another chromatid went on this side. So if you see here in mitosis, in anaphase, this chromosome and this chromosome, they are pairs of one another, so they are getting pulled, and this chromosome and this chromosome, they are pairs of one another, so they are also pulled to one pole. So in mitosis, pairs were getting distributed equally. So the chromosome distribution was in pairs, and the equal number of chromosomes as compared to the mother cell, the same number of chromosomes were getting inherited to the daughter cells. But that is not going to happen in meiosis. Let us see how, when we start the meiosis, we will understand that. So I hope that you have revised your mitosis section and you have understood mitosis. So if you have understood mitosis, remember that meiosis 2, which is there, it is same like that of mitosis. Only the difference is in meiosis 1, because meiosis 1 have some special steps, sorry, has some special steps where there is a reductional division of the chromosomes that is going to take place. That is half of the chromosome. If there is a pair of chromosome, one chromosome will go to one pole and the another chromosome will go to another pole. Not a pair, one single chromosome will go. And the another thing is that crossing over takes place between the chromosomes. Let us see what is crossing over. Recombination takes place between the chromosome, where the genes are exchanged, okay, from one chromosome to another chromosome, which is responsible for the combination of new genes, for the variations in the genes. So recombination, crossing over, synapses, okay, then chiasmata formation. So all these things are basically going to happen here, and that's why sexual reproduction, okay, when sexual reproduction happens, meiosis takes place for the gamete formation, and when the gametes are produced, these gametes are going to contain the combination of genes from the parental side. The same exact identical genes or chromosomes will not be inherited. Okay, so the combination of the genes takes place, the DNA exchange takes place, and that's why combinants are formed, recombinants are formed. That's why when the individuals are born, when the offsprings are born, these offsprings are not identical copies of their parents. These offsprings are basically a combination of characteristics from their parents. Okay, you must have seen that some characters are like that of father, some characters are like that of mother. Okay, paternal characters, maternal characters. Sometimes it is also seen that the characters that were present in forefathers, okay, in grandparent or great grandparents, those characters are also sometimes seen, they're expressed in the after 2 to 3 generations. So that is only because of the gametes which are produced with the help of meiosis, recombination and crossing over that happens in the meiosis. Now let us start with meiosis 1 only. Let us only understand about the meiosis 1 first, and especially in meiosis 1 the stages that we will study is prophase 1, okay, then there is metaphase 1, anaphase 1, and then there is a telophase 1. Okay, now here the karyokinesis takes place, that is a nuclear division takes place. But what is seen is that after telophase 1, again we have to compare this with mitosis. We studied that after the telophase gets completed, the cytokinesis follows and the cells are divided, okay, and the two daughter cells are formed. Here maybe the cytokinesis may take place. If not, suddenly the cell from the telophase can jump to the metaphase 2. Okay, now that is something further that we will discuss, but let us understand about the meiosis 1. Then when we will discuss about meiosis 2, okay, we have to study the phases like prophase 2, okay, then there will be metaphase 2, then there will be anaphase 2, and then there will be telophase 2, which will be followed by cytokinesis. And what we have to remember here is that once the complete meiosis process takes place, four daughter cells are formed, and all of these are going to be haploid in nature, right? So these are some of the things that we have to keep in mind when we talk about the meiosis. Now coming back to the prophase 1, okay, prophase 1 for our convenience to understand and to study and to write the answers, it is again further divided into so that it can be easier for us. Prophase 1 is divided as the leptotene stage. In some books it is given as leptonema, okay. Then there is zygotene stage, okay, some books it is given as zygonema. Then it enters into the next stage, that is a pachytene stage. Then it enters into the diplotene stage, and then it enters into the last stage that is called as the diakinesis. So these stages we have to study one by one. So then here the prophase 1 gets completed. Then the cell would enter into metaphase 1, anaphase 1, and the telophase 1. Right? So let us start the discussion with the first phase, that is the leptotene. Now the diagrams have been already I have mentioned over here. Okay, let us continue with the leptotene stage or the leptonema. Now in the Greek language, okay, in the Greek language, leptos word which is there, it means slender or thin, okay, whereas nema, it means thread. So slender threads are observable. Now in the prophase of mitosis, if you remember, there are two phases in the prophase. One is early prophase, late prophase, where the nuclear membrane starts disappearing, okay, the other organelles inside the cytoplasm, they start disappearing, the nucleolus disappears slowly, and the centrioles which would be there, they will travel towards the opposite poles, and they will start the production of the spindle fibers, right? But that yet it is not going to happen now in the leptotene stage. Let us see. The nuclear membrane which is there, the nuclear membrane is still intact, okay, it has not yet degenerated. Then the nucleolus is still intact, okay. The centrioles, okay, they are slowly moving towards the opposite pole, or they negligibly they will be moving towards the opposite pole, or they are just there only. And the condensation of the chromosomes has begun, okay. Now we can see here chromomere they have written, okay, in the picture. What is a chromomere now, right? So chromomere is that part, we can say, of the chromosome which contains the DNA, which contains the genes, okay, and still it is yet condensating. And this leptotin stage, when we talk about, the nuclear envelope is present, and these chromatin threads, they are basically attached to this nuclear membrane, okay, through the plate which is called as an attachment plate. Sorry, which is called as an attachment plate. Just a minute. That is called as an attachment plate, okay. And these chromatin fibers of the interphase nucleus, okay, they shorten and they get elongated, and the chromosomes, they become completely clear over here, okay. Now when we talk about the next point, the chromosomes are replicated, but the chromatids are not yet distinguishable over here due to the presence of nucleoprotein core between them. Okay, so it is a big statement that I have made. What I'm trying to say over here is that the chromatids which are there, okay, they still, we cannot see them clearly. They are still not visible, and there is a lot of nucleoprotein over here, okay, in them. So that's why they are not clearly visible. In many animal cell, it also appear as the bouquet stage. Okay, so where the chromatin threads are attached to the nuclear envelope from one side over here. Okay, so their structure might be like this, right? And one of the two centrioles, they will start moving towards the opposite pole, but very negligibly. And at the end of the stage of the prophase one, that is diakinesis, these two centrioles, they get separated from one another, and they travel towards, exactly towards the opposite pole, form the asters, and they will start the production of the microtubules, that is the spindle fibers. Okay, so centromere, so centrosomes, or we can say centrioles, still not have yet moved to the opposite pole. They have begun traveling towards the opposite poles. Okay, now let us understand about the aster and the astral rays. Okay, when we talk about the animal cell division, it is called as the amphiastral. Why it is called as amphiastral? Because the asters are involved, which are to the opposite poles, and they produce the spindle fibers, okay, from both the poles. That is called as amphiastral. But in the plant cell, it is seen as an astral condition because asters won't form. Now, what is an aster? Basically, the complete pair of the centrioles, okay, along with their spindle fibers, they are called as the asters. And what are aster rays? These microtubules which are originating from the centrioles, they are called as the aster rays. So the pair of centriole and the aster rays together, they are called as the aster. Okay, and in animal, it is the amphiastral type of division. So these are some of the things in the leptotin stage. Okay, now let us see the next stage, that is the zygote stage. Okay, what happens in the zygote stage is that till the leptotin gets over, okay, these chromatin threads, they condense and they form, they get into their structure, which we now will be calling them as the chromosome, and the chromosomes will pair with their partners. Okay, suppose if this is chromosome number 1, it will pair with its chromosome number 1 partner. If this is chromosome number 2, it will pair with its chromosome number 2 partner. So these chromosomes are called as the homologous chromosomes. Why they are called as homologous chromosomes? Because they acquire the same position, okay, and they have the same type of genes, okay, they have the same type of alleles which are present on them. So that's why they are called as a homologous chromosomes. Okay, Now in the zygote stage, when they are aligning in front of their partners, this process is called as the synapsis. This process is called as the synapsis. So the nuclear membrane is intact, okay? Still the centrioles are moving towards the opposite pole, not yet reached to the opposite pole. The other organelles are intact, okay? The chromatin threads have condensed, they have formed the homologous chromosomes, and along with this formation, here what is going to happen is that a nucleoprotein complex is also formed in between these two homologous chromosomes, so they can be kept together. Okay, which structure has been formed? A nucleoprotein structure. Threads have been formed in between them. Okay, so this structure which is formed, the complex which is formed, this will be now called as the synaptonemal complex. Okay, now when we talk about the homologous chromosome, these two chromosomes together, okay, this will be called as the bivalent. What is called as? It is called as bivalent, and it is a tetrad condition. Why tetrad? You can see here, this is one complete chromatid. Okay, this is the second chromatid, right? And they both have one centromere. So these two chromatids will be called as the sister chromatids. Okay, and you can also see that in this pair there are these, this is one chromatid and this is the another chromatid. So they are also held together by one centromere. So they will be called as a sister chromatids. So how many chromatids we can see? Four chromatids we can see. So that's why the condition is called as tetrad. And sorry, how many chromosomes we can see? Two. So it is called as a bivalent condition. Okay, so these are sister chromatids and these are the non-sister chromatids, and the synaptonemal complex is getting formed. The synapsis has already taken place. Now let us understand what is procentric synapsis and pro-terminal synapsis. Okay, what is procentric synapsis and what is pro-terminal synapsis? What is procentric? Okay, the synaptonemal complex starts from the centromere, okay, and it continues towards the ends. Okay, so it starts from the center and then it travel towards the end. This is called as a procentric synapsis. Okay, or we can say procentric synaptonemal complex formation. What is pro-terminal? Okay, pro-terminal means this will start from the ends. Just a minute. This will start from the ends and it will move towards the centromere. So this is called as a pro-terminal kind of a synapsis. Okay, and then there is one more type of synapsis which is called as intermediate type of synapsis. What is intermediate? At various places, okay, in between centromere and the ends, the complex is formed. That is the synaptonemal complex is formed. That is called as intermediate type of synapses. Now these all 3 points were given by the scientist Moses in the year 1956. Okay, Moses in the year 1956. Okay, now let us talk something about the synaptonemal complex. We know that the synaptonemal complex is formed between the homologous chromosomes, right? And it is a structure that is formed of nucleoprotein. Protein is one part and the nucleo means the nucleic acids are also there. So it is called as a nucleoprotein. Just a minute. All right. Now let us talk about the synaptonemal complex. Okay, this is basically ribonucleoprotein and it has a tripartite structure. Tripartite structure. Let me draw a representative diagram over here so then we can discuss on this. Okay, so this is a synaptonormal complex. Now how many elements that we can see here? Okay, these elements are called as the central elements. Okay, and these purple color which I have drawn, those are called as the lateral elements. Okay, central element is the element that is present between the homologous chromosomes. Let me draw here the homologous chromosomes. So it is present over here. Okay, that is a central element that is present over here. Where is the lateral element present? Lateral element is present between the sister chromatids somewhere over here. Okay, so this is how the this is how the synaptonormal complex appears, and along with that there is this roundish purple color structure that we can see. This will be called as the recombination nodule. Recombination nodule, which will be present at certain specific regions over here. So what is this recombination nodule? Basically, this recombination nodule has different types of enzymes. Okay, enzymes like recombinase enzyme. This recombinase enzyme again has different types of enzymes we will study now in the next stage, that is the packaging stage. This recombinase enzyme helps in the process of recombination. Okay, recombination of the chromosome segments, okay, which are transported from one chromatid to another chromatid. But this is all about the zygote over here. Okay, so in zygote what we have seen, just let us have a recap, that the synapsis takes place, that is the homologous chromosomes, they pair up, synaptonormal complex forms. That is what is going to happen. We saw that how what is a synaptonormal complex. It is a nucleoprotein complex, and there are two elements. One is central element and the another are the lateral elements. Central elements are present in between the two homologous chromosomes, and the lateral elements are present between the chromatids of the same chromosome. Right, and there is a recombination nodule. So now what has happened is that these two chromosomes, homologous chromosomes, are now patched up with one another, so that the next stage, which is the packaging stage, and the crossing over and the recombination can happen successfully. Okay, now let us move on to the next phase, that is the packaging stage. Okay, packaging, or it is also called as a pachynemma. Okay, pachys means thick. Okay, and tenia means band. Okay, so thick bands are going to get formed. The diagram is over here. Now what we can see here in the diagram is that as soon as the synapsis is over, okay, the cell is going to enter into the packaging stage. Right, now the cell can remain in this stage for many days, not only for few hours but for many days. Okay, and chromosomes are paired and they occur in a synaptonormal complexes, we have already studied. The paired chromosomes are bivalent, we have already studied, and they shorten up over here. Their length gets shortened, they become thick. Okay, now we have already studied that each bivalent is made up of four chromatids, right? So here are the four chromatids that we can already see over here, right? Now dense areas Okay, dense means very thick and dense areas appear on the chromatids over here, on the bivalents over here, which are called as the recombination nodule. We have just studied in the previous zygotine stage that the recombination nodules were present. Now these recombination nodules, as I told you, has one enzyme which is called as a recombinase enzyme. Okay, recombinase is basically made up of, it is made up of endonuclease enzyme, it is made up of exonuclease enzyme, it is made up of an enzyme which is called as the unwindase enzyme, and R protein, etc. Okay, in the presence of endonuclease, what does the endonuclease enzyme does? Now basically why this process is happening, if you see over here, this is one chromosome and this is the another chromosome. Okay, what we can see here is that here the two non-sister chromatids have overlapped on one another. What has happened? The two non-sister chromatids have overlapped on one another. Now this is called as overlapping or crossing over, and there is one point, okay, below that, below this chromatid arm, there is one point where both of them have got attached to one another. This region where they are attached to one another is called as a chiasmata formation. It is called as the chiasmata. Now these endonuclease enzyme, what it does is that for our convenience, what we will do is that we will take chromatid arms which are of different colors, or we can see here directly in the diagram itself. This chromosome is blue in color and the another, its pair, is pink in color. What is happening over here is that you can see there is a pink structure up till here and afterwards it has become blue. Okay, and this blue chromosome, after some structure, after some length, it has become pink in color. Means what is happening, there is exchange of chromosome segments that are going over here. Okay, some regions, some segments of each of the chromatid are exchanged with one another. So exchanging means obviously the chromosome contain the DNA molecule on it. DNA short segments are called as genes, means there is the swapping or there is an exchange of genes that is happening between these two non-sister chromatids. And for that purpose, to cut the chromosomes or to cut the chromatid at specific site, which is called as a nicking, okay, this endonuclease enzyme is required. Exonuclease, it is going to cut the DNA biomolecule on its outer side. Endonuclease is going to cut the DNA molecule on its inner side, somewhere on the inner side. So that's why it's called as endo. Endo means inner side, exo means on the outer side. Unwindase, what does the unwindase enzyme does? It unwinds the DNA molecule for the cutting purpose. Okay, so that is the unwindase enzyme. What is the role of R protein over here? Once these segments of the chromosomes have been transferred from one another, okay, that patching has to be done. The reunited phenomenon has to happen. They should be reunited with one another, right? So for this enzyme, the R protein, it reunites the cut segmented portion with the chromatid arms. Okay, so that is the R protein, and this reunited phenomenon that is happening, it is called as the annealing process. Okay, so here basically a crossing over phenomenon is taking place. We have to remember this, right? The two non-sister chromatids, they overlap on one another, they cross over, and once the crossing over has taken place, the chiasmata formation takes place. They get attached to one another certain specific region, and the recombinase carries out its role. It cut the segments of the chromosomes and it exchanges. Because of this, the exchange of the chromosome segments takes place, the DNA gets exchanged, the genes get exchanged, and in this way, the new types of chromosomes are now formed, which are called as recombinants, right? So because of the recombination process, variation of genes takes place. Okay, new types of gene, sorry, new types of gene combinations are formed which is because of which the evolution process takes place. Okay, so for the process of evolution, the recombination and the variation has to happen, then only the evolution can continue, right? So this is all about the packaging stage. I hope we are clear about the packaging stage. Now the second last stage that we will discuss is about the diplotin stage. Now diplotin stage has very few points in this. You can see in diplotin stage, okay, now the crossing over has finally done, the recombination has finally done, it is successful. The synaptonormal complex, okay, in the diplotin stage, the synaptonemal complex which was formed during the zygote, it gets completely dissolved. Okay, now there is no synaptonemal complex and the chiasmata which has formed, this chiasmata only is present. That is, the two non-sister chromatids are attached at one point, but there is a dissolution of the nucleoprotein complex, that is the synaptonormal complex, that has taken place over here. Okay, however, it is not a permanent structure. They shift sideways, okay, and also they might disappear. So that is what is going to happen at the end, but only the chiasmata we will see. So it will be something like this. The synaptonormal complex is now not present and only the chiasmata is present. The remaining structure has been completely dissolved. Okay. It is also seen that in the vertebrate oocytes, it is seen that many vertebrate oocytes are in an arrested diplotin stage for many years. They can remain in this arrested stage for many years. The chapter of human reproduction, and when we talk about the oogenesis, okay, it is seen that the primary the primary oocytes which are formed, they get arrested in the prophase 1, okay, of the meiosis 1. So which is that stage? If you're in, if you are studying in second year and if you have biology, you must have studied that point. So that the primary oocyte is arrested in the prophase 1, but exactly where it is arrested, it is arrested in the diplotin stage. Okay, so the oocytes are arrested in the diplotin stage, a very important point. Another thing, here it is also seen that there are some chromosomes which are called as the lamp brush chromosomes. Now, where are these lamp brush chromosomes? Over here we can see the diagram over here. These are the lamp brush chromosomes. Okay, so diplotin is extended, okay, for many years. They are present in this form only, and metabolically it is active in animal cells, okay, especially oocytes, which we have already just discussed. And in such cases, what also happens is that because they are arrested in such a, they are arrested in this stage for a longer period of time, the chromosomes, they may get decondensed, right? The chromosomes, they may get decondensed, and the DNA which is there, it gets involved in the formation of RNA. And when the RNA are formed, we know that the proteins are also synthesized. So such kind of chromosomes are called as the lamp brush chromosomes. Only the thing that we have to remember is that the lamp brush chromosomes, what are they? They are those chromosomes which are arrested in the diplotin stage and they have got decondensed. Okay, so this is in case of the lamp brush chromosomes. And the last stage now that we will talk about is the dikinesis. And here in the dikinesis, the prophase 1 is going to get over. So what we can see in dikinesis, dia, it means through, okay, and kinesis, that is a movement, okay, that is a movement. So chiasmata, which we just have studied, okay, the chiasmata starts shifting towards the ends of the chromosome. Means, suppose if the chiasmata is over here, this chromosome, then it will come over here, then they will get detached from one another. Okay, so the chiasmata comes to the end, okay, and the chromosomes finally get detached from one another. This phenomenon is called as the terminalization of the chiasmata, we can say, or the terminalization of the non-sister chromatids that happens in the dikinesis. Okay, now here, if we see, the dikinesis is over, but now the cell is ready to enter into the next stage, which is called as the metaphase 1. Okay, now as the metaphase has to begin, we have studied in mitosis that in metaphase all the chromosomes they align in one plate, okay, and that is called as they align in the same equatorial plate. But for this purpose, to pull all the chromosomes in the same equatorial plate, the spindle fibers are required. So now till these stages were getting completed, the centrioles must have traveled towards the opposite poles, and the centrioles, they have started the production of the spindle fibers. Okay, and these spindle fibers, they travel towards the chromosome that we can see over here, and these chromosomes they get attached to the spindle fibers. But here we have to remember that in metaphase 1, now suppose if this is a pair of chromosome, okay, what was happening in mitosis? In the starting of the video lecture, I have told that in mitosis, okay, the spindle fiber comes and attaches from both the sides to a single chromosome, right? Here it is not like this. The spindle fibers from one pole will attach to one member of that pair, and the spindle fiber from another pole attaches to the another member of that pair. Okay, so they have been arranged like this, right? So now in at the end of the dikinesis, all the chromosomes have been attached with the spindle fibers, and at their centromere, okay, there are kinetochores, and the kinetochore they bind to the spindle fibers. All these are pulled to the same equatorial plate, right? And this is about the metaphase 1 that will be then completed. And once the metaphase 1 is completed, then starts the anaphase 1. Now, why they have been brought to the same equatorial plate? So that they can be divided properly. Now, here the reduction division has to take place, right? The half the number of chromosome has to be divided. So, here what is going to happen in anaphase 1 is that now this is a pair, okay, we call it as a bivalent and we call it as a tetrad because this pair had 4 chromatids. This one chromosome will be pulled towards one pole, and this one chromosome will be pulled towards another pole, right? So what is happening here? Okay, a pair of chromosome is not equally divided now. Okay, instead the pair is getting reduced. So that's why this is the reductional division. So where does exact reduction of the chromosome happens? It happens in anaphase 1. Okay, in meiosis 1, in prophase, in meiosis 1, in anaphase 1, the exact reductional division takes place. Where does the crossing over takes place? It happens in the Pachytene stage. Where does the synapsis takes place? It happens in the zygotine stage. Where does the terminalization takes place? It happens in the dikinesis stage. Okay, so these are some of the things you can expect the questions on. So now let us compare this with the anaphase of mitosis. Okay, some of you might be wondering what is happening over here, how this is reductional division. Let us see here. Okay, in anaphase in metaphase of mitosis, okay, this is a pair of chromosome. But what happened over there? The spindle fiber attached from this side and the spindle fiber also attached from this side to this chromosome. Here also it happened attached from this side. So what happened here in the anaphase 1, the centromere splits, right? And as the centromere splits, the chromatids they get separated from one another, right? So this will be pulled on this pole. This is also going to be pulled on this pole. So here a pair of chromosomes are traveling towards the opposite pole. This will be pulled to one pole and this is also going to be pulled to one pole. So here also a pair of chromosomes are traveling towards the opposite pole. So when the cell will be formed, when the daughter cell, see, the daughter cell is having a pair of chromosomes, but that is not the case over here in anaphase 1. In anaphase 1, Still, the half of the division has taken place over here. We have to still study metaphase. We have to still study meiosis too. But here, this one chromosome is pulled towards one pole and the another chromosome is pulled towards another pole. So now the pairs are not present. They have been separated from one another. I hope this is clear. Let us continue with the next phase, that is about the telophase 1. Okay, now one chromosome is at this pole, another chromosome is at this pole. So what will be calling this? We are going to be calling this as a dyad. Okay, because how many chromatids we can see? Two chromatids we can see, which are connected to one another with the help of one centromere. So this is now the dyad structure. Okay, so the nucleolus is formed over here. It is also followed by the appearance of the nucleoplasm, and the nuclear envelope is also formed over here. Okay, the nucleolus is also formed over here. Okay, the elongated chromosomes usually remain straight, and now they are not going to enter into the interphase. Okay, so in mitosis they could have entered into the interphase after cytokinesis, right? But here they are going to remain like this. In some cases, I've also told you that telophase, from this telophase 1 they can directly enter into the sorry, from the anaphase only. Let me revise it. Okay, from the anaphase they will skip the telophase. Okay, and sometimes in some cells, when the anaphase is over, they will skip the telophase and they will enter into the meiosis 2. That is, they will directly enter into the prophase 2. Okay, but for our convenience, we will first understand what happens in the telophase. We have seen what happens in the telophase. The nuclear membrane has disappeared, the nucleolus has reappeared, and when we talked about this scenario, that is anaphase, from the anaphase the telophase is skipped and it enters into the meiosis 2. This happens in a plant which is called as trillium plant. Okay, so this is all about the meiosis 1. We have completed till the telophase 1. In the next video lecture, I will discuss about the meiosis 2. So I hope this is clear, and we can see here that only one chromosome is present. It is not a pair, it is a single chromosome. So the reduction and division has taken place. And then in the next video lecture, we will discuss about the meiosis 2. Thank you so much everyone for watching.